let's begin our second lecture on membrane structure and function. In concept 7.2, we're going to talk about membrane structures that result in selective permeability. By now you should have some idea of the fact that this membrane is a fluid and complex environment and that the proteins embedded into that membrane are important for performing functions. But the nature of that membrane, with hydrophilic outsides and hydrophobic inside, helps to provide what we call selective permeability. Permeable means that things can pass through, but that selectivity says that only some things can pass through. Hydrophobic nonpolar molecules like hydrocarbons and inorganic gases can dissolve in the lipid bilayer and diffuse through. Hydrophilic molecules like ions and polar molecules, on the other hand, do not cross the membrane easily. Remember, they can interact with the surfaces of the membrane, but they're not going to pass through that hydrophobic core. Instead, proteins that are built into that membrane play key roles in regulating that transport. Transport proteins allow the passage of hydrophilic substances across the membrane. Some of these are called channel proteins because they are quite literally a channel between the hydrophilic sides of that plasma membrane. And so certain molecules or ions can use that channel. One example of this is the aquaporin channel protein, which we saw in our first section of the lecture. Aquaporin, as aqua might suggest, allows the passage of water molecules. It does so at a much faster rate than water, which is a hydrophilic molecule, would naturally cross the membrane. So it's much more efficient at maintaining that important balance when needed. Carrier proteins are a special kind of protein that can bind to molecules and actually change shape to shuttle them across. Here are examples of what these look like. Diffusion is a natural process where a molecule can pass between the two sides of that phospholipid bilayer. It doesn't need the help of a protein. It may not be very quick, but it's going to occur on its own. Other proteins, like channel proteins and carrier proteins, can allow the transport from one side to the other. It's important to note that in all of these three cases on the left side, we are moving from high numbers of molecules to a space with low number of molecules. Keep that in mind for later. Another kind of transport, like the one you see on the right, is actually going to move these molecules in opposite directions. They're going to move molecules from low concentrations into high. Regardless of this, all of the proteins are still classified as transport proteins, whether channel or carrier proteins, passive or active. We're going to begin with a discussion of passive transport. This includes the diffusion of a substance across a membrane where no energy is invested. And diffusion, simple diffusion, is probably the easiest. There's a tendency for molecules to spread out evenly to fill unavailable space. Kind of like how when people enter into a room, they naturally spread out. Although each molecule moves randomly, there can be a semi-directionality, where if one side of the room is very empty, a lot of the people will tend to fill that space and create equal spaces between various other people in the room, giving everyone equal space around themselves. There's what we call a dynamic equilibrium, which you can see on the right, where the molecules are moving in such a fashion that they're filling out both sides equally. It doesn't mean that they're not moving from one side to the other, but as fast as they move from one side, another molecule moves in the other direction. Note that if you have two different molecules in a solution, they don't all have to be moving in the same direction. Each solute, each molecule that is dissolved, is going to have its own diffusion rate, and it's going to have its own equilibrium. So you can have a variety of different molecules that are trying to cross this membrane at the same time, sometimes even in different directions. Always, though, they're trying to reach their own net equilibrium. There are some additional videos for those of you that would like to see some of these in action, including diffusion and a section about that membrane selectivity we've talked about. I want to give a name, though, to the direction in which molecules tend to diffuse. We call this a concentration gradient, where the concentration gradient goes from more dense to less dense, and the fact that molecules, solutes, tend to move down the concentration gradient to areas of lower density. Because this occurs naturally, the cell doesn't have to expend any energy to be able to achieve this. And so we call this passive transport because the cell can simply allow it to happen passively, no energy needed. A great example of this is osmosis. Osmosis is simply the term we give for the diffusion of water across a selectively permeable membrane. As you know, water is very special, so sometimes it gets its own name for the same thing. Water is going to diffuse across a membrane from the region that has low amounts of solute to the region that has high amounts of solute. We can explain this by the fact that water that is forming hydration shells around these solutes isn't really free to cross the membrane. The only water that's going to be moving through that selectively permeable membrane, the only water that's actually going to be influencing that dynamic change, is not going to be interacting with solutes. And so we only account for those that are not interacting. And so if you have a section with a lot of dissolved solutes, you have to move warm water molecules to that side in order to further dilute out and have equal numbers of free molecules. Here's another picture to give you a sense of that and to remind you of what these hydration shells actually mean. We use the term tonicity. This is the ability of a surrounding solution to cause a cell to gain or lose water. Same principle as osmosis. We're just going to give it a name to describe how this solution influences the osmosis of a cell. An isotonic solution is going to have a solute concentration the same as the cell. This means no net movement across the membrane. Hypertonic solutions are going to have a solute concentration that is greater than inside the cell. So you would have more on the outside, which means you're going to have more hydration shells. Those hydration shells are going to use up the water molecules, and so water from inside of the cell can pass through the membrane to try and replenish that water concentration outside and cause the inside and outside to become equal. Hypotonic solutions have a solute concentration that's less than the inside of the cell. What's going to happen here is that because there's fewer hydration shells on the outside, you're going to have more free water, and so it's going to rush into the cell in order to be able to even out those concentrations of free water. So what does this look like? Well, on the left we have that hypotonic, where you have water rushing into the cell. Remember, you have very little solute on the outside, versus hypertonic, where you have lots on the outside, and so the water is going to be rushing out. In like a red blood cell or a lot of other animal cells that don't have cell walls, this is easily going to cause disruptions to the structure of the cell. In hypotonic solutions, the cell will swell or even burst, and in hypertonic solutions, it will shrivel. This is based on the fact that the water is going to be leaving or entering the cells, but not in equilibrium like in an isotonic solution. Plant cells and other cells that have cell walls are going to have a bit of an advantage. The cell wall is going to contain some of that shape. So, for example, when you have a plant cell in a hypotonic solution, although it is going to swell up, which we call turgid, it won't burst the way that an animal cell without a cell wall might, because there's going to be much more support. In a hypertonic solution, this can actually cause so much water loss, though, that you begin to see the plasma membrane move away from that structured cell wall. In the middle is what's known as flaccid, and you can imagine what this might look like in a plant. Now, these environments create problems for organisms, especially as highly hypertonic or highly hypotonic environments that have cells without rigid walls. Osmoregulation, remember, the movement of water is called osmosis. Osmoregulation is going to be the control of solute concentrations and water balance, and it's a necessary adaptation for life in such environments. So, for example, we have seen a contractile vacuole in action. This is necessary for the eukaryote Paramecium, which in its hypertonic environment has to be able to pump water out of the cells as fast as it rushes in. In plants, we can see this, remember, they have cell walls, changing the structure of the plant. To better show this, I'd like to give you some videos. Plasmolysis is going to be caused when the cells are dried out because there's so much solute on the outside, so many hydration shells, that the water rushes outward. You can see the plasma membrane actually pulling away from the cell walls. In the opposite, these same plant and its cells are becoming turgid, where they're actually swelling and growing and filling up all available space within the strictures of that plant cell wall. Note how cool it is to see all those chloroplasts moving around in live cells, because they're just naturally green and easy to see by light microscopy. Facilitated diffusion can be both through a simple diffusion, a natural diffusion, or facilitated by proteins. So this natural diffusion is going to occur based on that chemical gradient. But facilitated diffusion will use that same gradient at a faster pace. So it's still passive because no ATP will be used, but proteins will make this happen faster by overcoming some of the challenges of the natural properties of the plasma membrane. Channel proteins simply provide corridors that allow a specific molecule or ion to cross the membrane, like the aquaporin that facilitates the diffusion of water. Ion channels are another form of these channel proteins that specifically allow ions in and out. Some of those ion channels will also be what we call gated, where they can actually decide, based on the stimuli around them, whether or not to allow those ions to come in or out. So it's like a channel protein that can be on or off depending on the needs of the cell. Carrier proteins can have subtle changes in shape. These allow the protein to take up from one side and dump it on the other. This is a little different from a channel protein, which simply provides that electrophilic or otherwise perhaps charged space to facilitate diffusion. These actually have a mechanical movement. Please note, though, that we are still moving down the concentration gradient, and so you do not have to export any energy in order to do this. Here's an example of a passive carrier protein. This is GLUT1. Note the sequence of the amino acids in the first diagram. Each one of those little circles represents an amino acid with its own individual variable group. They've been shown here in secondary structure, forming alpha helices, which you can now see embedded as transmembrane domains within the plasma membrane. This is encoded by the gene SCL2A1. You don't have to know any of this, by the way. That gene, which is found in the DNA of your nucleus, gets copied into an RNA, transported out into the cytoplasm, and then on that rough ER is translated into protein by a ribosome, where it forms the secondary alpha helice structures due to hydrogen bonding between the carbons, or the carboxyl and the amino groups, and embeds in the membrane. Eventually, these alpha helices and the variable groups of various different amino acids within the chain will further interact into a tertiary structure, which is shown in the red ribbon diagram. Note that it has some dotted lines that show you the portion which is still embedded in the membrane. So, here is our transmembrane protein. It's a carrier protein that transports glucose across the membrane. It's highly specific. Even fructose will not be passed. Remember, it's a selectively permeable membrane, and proteins like this can help to achieve it. This is just one example of a glucose transporter, and there are lots of different ones. This is just an example to show you how scientists have worked out this movement. Note that when you have the ligand bound, you get changes in these little X's. They represent those alpha helices that you saw, and so they can actually open one side or the other to allow a glucose molecule to pass through. That's what makes this a carrier protein, not a channel protein, is that movement. So I would like to know, GLUT1 is a transmembrane carrier protein responsible for the passage of glucose into cells. It's highly expressed in fetal and brain tissue. Genetic defects in GLUT1, this is known as Vivo disease, would produce which of the following symptoms? Type 1 diabetes, small fetal brains, we call this microcephaly, weakened muscle state, low intelligence, or all of the above. The answer is microcephaly. If you answered type 1 diabetes, great job for thinking about the importance of glucose in a disease that we've already touched on. But because this particular carrier protein is expressed in fetal and brain tissue, it's not going to have a huge global impact on blood sugar levels. Similarly, when we discussed a weakened muscle state, this protein is not found on muscle cells, so I wouldn't expect it to influence that energy producing ability of your muscles. So that leaves us with either small fetal brains or low intelligence. Although it is true that children with Vivo disease do have low intelligence, this is due to the fact that they have very small brains. Expressed in fetal tissue means that there's very little energy available to a growing fetus, with a lot of cells growing and dividing. And especially in the brain, where this protein becomes found in high concentrations, there simply is not enough energy to sustain a growing human being and its very large brain. The result is a small brain, which we call microcephaly. We'll continue this in our next video, Lecture 6.3.